Well, hey there, friends. This is our final discussion on genomics and things, talking a little bit more about repeat sequences and what they are, how they are generated potentially. In terms of what they do, not a whole lot of clue, but we were mentioning yesterday, or last time we talked, about the idea of gene families. The fact that when you begin looking at the genome, you can find genes that have significant amino acid and or nucleotide similarities, identities. And they're clearly not the same gene. They may have widely different expression patterns, but they do have some homology to them. We mentioned the example of G protein coupled receptors, and this is a family of receptors that are really based on a standard kind of structure, a 7 membrane pass protein. And we'll be looking here. Globin genes are another gene family. There's the actin gene family, myosin gene family. So these are genes that share very similarities in terms of structure and amino acid sequence, but they're not identical. And they're going to have different kinds of developmental timings. They have different functions in terms of, like as an example, myosin is a contractile protein that binds to actin, but you got muscle myosin, and then you have cytoplasmic myosin that's involved in movement of things in the cytoplasm and the production of the contractile ring. They're clearly very similar, but they're not the identical gene. So this is an example of some of the kinds of repeats that we see. And it is hypothesized, and probably a reasonable hypothesis, that they were derived by duplication of ancestral genes, and then you get this accumulation of mutations or changes between the copies. So, another term that is found, or an idea that's found in the genome, is this notion of what's called a pseudogene. And pseudogenes are genes that you can have a very clear homology with, but they are typically inactive. They will have a stop codon very close to the front end. They'll have a splice junction mutation. They'll have maybe some kind of problem with the promoter sequence. These are inactive, but they're present in the genome, and they are not functional. So they usually are being blocked through transcription or translation, or both. And another example of the fact that there are these remnants of gene replications. Another thing that gives us information about this notion of gene replication is the idea of gene clusters. There are, in the region of the genome, you can find groups of adjacent genes that are very similar. An example is a set of transcription factors called the homeotic genes. These are involved in, well, they were first discovered in flies, but they're also found in vertebrates, and they're involved in determining specific kinds of pattern formation and specific kinds of identities of different structures. In the fly, it's the difference between having an antenna versus a leg. It is a single gene event. And homeotic genes are also found in vertebrates. They're involved in the patterning and differentiation of the spine, for example, and actually in a variety of different places. They're very clearly similar genes, but they've got slight differences, and specifically with the case of the homeotics, their primary difference is going to be the DNA binding domain. These are transcription factors, and so they bind slightly different sequences. So again, replication going on, and this is actually in tandem. It's groups of genes in a row. So it is believed that this kind of recombination and duplication occurs primarily through recombination and unequal crossing over. So if you recall back in genetics, discussions of recombination, let me get a little pointer here, beep, 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 there. We can have, during meiosis, the interaction of two chromatids, and they actually can have an exact break that results in a chromosome fusion that is in fact a nucleotide exact precise break there, and this results in a recombination of the two different chromosomes. So the recombination actually has a base for base exact match. Now, what can happen though is unequal crossing over. And in this case, unequal crossing over is going to happen because you have a mismatch in the exact spot where the recombination should be occurring. And so one chromosome is going to end up losing and one chromosome is going to end up gaining. And unequal crossing over can generate a series of repeats. And this is a pretty good model for how repeat sequences are being generated. Now, we can repeat large pieces, genes, and we can develop over ancestral evolutionary time gene families, but another aspect of the genome is a series of very small repeats that aren't part of genes. And for a variety of reasons, these have been identified pretty much by accident. And they're referred to as satellite sequences, satellite DNA. And this is DNA that consists of a bunch of tandem repeats, all identical, and they are made up of a very short basic repeating unit, and they can be thousands and thousands of repeats. And so you may have a 6-nucleotide repeat over and over and over again. It's referred to as a satellite because one of the things that you would observe is that doing a restriction digest of specific, using specific enzymes, you can end up cutting within a repeat and generating a band that is very distinct among this smear of DNA fragments that you generate when you digest a whole genome. And so doing what's called density gradient centrifugation, where you put DNA into a tube with a very dense salt solution, cesium chloride, and you spin it at ultra centrifuge speeds, you end up generating a gradient of cesium chloride, and DNA will migrate to a specific spot in there. The genomic DNA will generally migrate to a very specific spot, and then there'll be bands that are below it, referred to as satellites, that are smaller fragments, but they are consistent because these are all repeat sequences. So once we got satellites, then people began looking at them more carefully, and you have mini satellites, and then you even have microsatellites. So mini satellites are tens of base pairs, and then we have microsatellites that are even smaller. And unequal crossing over is going to be responsible for actually, in one generation, causing an increase in the copy number or a decrease in the copy number. So unequal crossing over can do some actual significant things. One of the things that's been studied back in the 70s, even probably before then, is conditions known as thalassemias, which are caused by deletions of different globin genes. You may not know, but there are multiple globin genes in the human genome. And these are the genes that are responsible for carrying oxygen in red blood cells. So, just not to get too involved in the physiology, but if you think about this, when you're a fetus, your globin actually needs to pull oxygen from mom's globin in the placenta. So it actually has to have a higher oxygen affinity. And it's able to pull it out. It's not being oxygenated through the lungs. It's actually being oxygenated by touching the blood. As when the baby is born, there is a shift over the next couple of weeks of the gene expression to where you now begin producing the appropriate globin complex. And so thalassemias result from deletions of different alpha globin genes. So these have been studied because you end up with babies and fetuses that are in oxygen distress. And so people begin doing various analyses on the deletions in these genomes. And there's a range of different kinds of diseases, disproportionate amount of the beta-4 tetramer relative to the amount of the normal hemoglobin alpha-2 beta-2. Some of these things are actually fatal because you're missing the alpha gene. So thalassemias result because of the previous gene duplication events actually undergoing deletions. All right. There's even fusions of different globin genes and things as well. So There are other repeat sequences as well. One that we talked a little bit about when we're looking at COT curves is the ribosomal RNA genes. These are a series of sequences that encode an RNA that is used in the construction of the ribosome. So the ribosome, remember, is a RNA and protein conglomerate, a ribonuclear protein. And the ribosome structure comes from the RNA, and so you have a lot of ribosomal RNA. And the genes in the ribosomal DNA cluster are all identical sequences. And so you have so many of these ribosomal RNA genes in tandem, and they're being expressed at a high level, that you actually generate a slight weird discontinuity bubble in the nucleus. This, of course, is the nucleolus. So one of the things that's interesting to look at is that between the individual genes is the non-transcribed spacer. And that actually can have a fair amount of divergence to it. And it's actually sometimes used as a identification tool for identifying different species. So the non-transcribed spacer actually can have a wide variety of sequence and things. It's not actually part of the duplication. So, as we say, we have the nucleolus. Let's see. Yeah, nothing all that exciting here. It's just that various ways of determining these things. Now, another event that can happen, and this is showing unequal recombination, is allowing for the advancement or contraction of a repeat sequence. And there actually are some diseases that are generated through this. Huntington's disease is one specific repeat sequence disease. So the protein that's responsible for Huntington's disease is called huntingtin. The gene has something known as a polyglutamine or polyQ repeat, in that there is a whole set of the codon CAG coding for glutamine, and there'll be a bunch of them. Now, if you've got less than around 10 or so, you actually are lethal. You can't survive. That protein won't function properly. Most people have somewhere between 10 and, say, 22, and that's going to be just fine. Some people begin to have repeat sequences that are going over 25, up into 30, and even sometimes into 40s repeat sequences. If you're too long in your repeat sequence, you actually can, over time, begin to exhibit neurodegenerative defects. And so Huntington's disease is a neurodegenerative disease. And one of the things that has been observed with Huntington's disease is that it exhibits something called anticipation, in which grandfather might show symptoms of Huntington's at age 80. The son shows symptoms of Huntington's at age 50, and the grandson might show symptoms of Huntington's at age 30. And what this has actually been determined to be is that over the course of these different generations, the polyglutamine repeat has expanded. And so people actually can go and look at the number of polyglutamine repeats that an individual has and make an estimation of, are you within the normal range, or are you at risk of having Huntington's disease, and how early might it occur? So it's actually an interesting kind of phenomenon that is involved with recombination and things. Let's see. Another part of satellite sequences in these highly repetitive DNAs is they generally are on portions of the chromosome known as heterochromatin. And heterochromatin, we haven't really talked much about this, and we're not going to talk much about chromosomes because it's kind of boring. Chromosomes are defined by their compactness, and whether they actually have genes within them that are coding. And regions of the chromosome that don't contain transcribed genes are often very, very compacted. And they are referred to as heterochromatic sequences. Sequences in the chromosome that are being transcribed end up being a bit more open, and those are referred to as euchromatic sequences. So when you look at a chromosome, and if you've seen these like chromosome maps, and I'm trying to think what it's called, a Miller spread? No, anyway, when you do a a chromosome analysis of most eukaryotes, what you do is you block cell division, and you block it at the spot metaphase, and so you can't get past metaphase. And so the chromosomes have condensed, and you then drop them onto or spin them onto a little microscope slide, put it on a scope, and then a cytologist can recognize the individual chromosomes. And the chromosomes exhibit a banding pattern where you have these darker areas, which are the heterochromatic regions, and the lighter areas, which are the euchromatic regions. And it has to do with DNA packing. The point being, satellite sequences are often found in these heterochromatic regions. And in part, these are providing structure to the chromosome. So the spot where the spindle fiber binds, that's going to be a very intensely heterochromatic, highly repeated sequence. So regions of the chromosome that are actually coding for genes are going to have a slightly different look to them, and they are part of this euchromatic sequences. Let's see, is there anything else here? Yep, in situ hybridization by looking at squashes on a microscope slide. And what you can look for and identify is regions of mouse satellite DNA. In this specific figure here, we're looking at repeat sequences that are actually associated with the centromere. And this is an example of what we talk about, that these structural locations where the spindle fibers are going to bind. There's no genes there. It is just DNA sequences and these repeats, and it's very tightly bound. And that makes sense. All right. Let's see. Anything else interesting here? Nah. Mouse satellite DNAs, million satellite DNAs, and then you can begin looking at consensus sequencing things. Oh, all right, so here is actually something that is of interest to me, is that microsatellites can vary within individuals, and they can be used as determinants of identity. Like you can do paternity tests and things. And one of the systems that is used is something that's known as a VNTR, or a variable number tandem repeat. And these are very short, usually 6, 3, 5, or 4 nucleotides repeated over and over again. And these are part of this system of identified sequences in the genome. So VNTRs can range from either being microsatellites or mini-satellites. We work with VNTRs that were what we would consider microsatellites. They're like 18 nucleotide repeats. Well, that brings us to the idea of DNA fingerprinting. And in fact, it's one of the things that we've been working on in the lab a little bit, and that is being able to resolve different kinds of repeat sequences. Is there anything else here? Beep, beep. No, it's good. So let me just show you what we've been doing, or talking about what we've been doing. Lucy Koger will be doing a seminar on this, I hope. As some of you know, Alyssa, hi there. We do forensics, and I've been part of that, where we've been talking about DNA fingerprinting in a forensics setting. And if you ever watch any crime shows and things, you perhaps know of this idea, what's known as the CODIS DNA sequence or DNA database. CODIS is the Combined DNA Identification System. And it's made up of, around maybe 13, it could be 20 different sites scattered along the chromosomes. And what you got with CODIS is a series of essentially variable number tandem repeats. A whole series of identified repeats. And the reason this works is that you'll have a series of repeats within a spot. And this works because the flanking regions are not going to be changing within the population. They are essentially invariant. And consequently, we can use these flanking regions as primer sites for PCR. And so what we do is have PCR primers that are running and hybridizing to the flanking region, and then you run PCR, and you then are left with small fragments. Now, these fragments can change depending on the number of repeats. Now, the difficulty is the CODIS alleles that have been chosen are pretty small repeat sequences, and often the DNA that you generate, one of the CODIS sequences we're working with, you'll generate a 300 nucleotide or so DNA fragment. And then depending on the number of repeats, it might range to 326, or it might go to 284. I'm just making these numbers up. But you have a range of sizes. Now the difficulty is we're only talking about, in this case, maybe a 10% change in the nucleotide fragment size, and that can be very difficult to resolve. That's one of the things that Lucy and I have been working on, is different techniques for resolving. The standard way that CODIS alleles are resolved is through using essentially a DNA sequencing machine where you're using capillary electrophoresis. And this allows for a very precise quantitation of the exact nucleotide length. Now, what people have done is analyze within the population the frequency of repeat numbers. And so what you have then is at each CODIS site, you have the possibility of two alleles, one from mom, one from dad. And they could have two different repeat sequence numbers. What is the frequency within the population? That's what you begin to actually start tallying up, and that's what you can begin to generate this probability that somebody else would have the identical DNA profile. And when you're looking at multiple CODIS alleles, multiple spots, and you begin to actually do the statistics on this, very quickly you end up with numbers like 1 in a trillion possibility of somebody else having that exact CODIS allele profile. And that's how we're able to exclude individuals from being part of the DNA identification. I mean, pretty quickly with CODIS analysis, with really even just using a handful of alleles, not all 13 of them, you can generate numbers like 1 in a trillion. There have not been that many Homo sapiens on the planet in existence. So, yeah, it's pretty much a dead, yep, this is the identity. You then get into issues of evidence collection and chain of evidence, but in terms of the population statistics, this stuff is pretty rock solid. So that is variable number tandem repeats. Now, if you recall, in Molec and Cell, and we kind of, those of you who weren't in Molec and Cell, it's all right, because at least those of you that were there last spring, we never got around to really finish this thing up. We were looking at something known as an ALU repeat. It's essentially an ALU insertion. And so ALU repeats are all over the genome, and they typically are a single fragment that can be present or absent. So within the genome, they're repeated in a single gene. You're going to either have one or you're not. And these also have the same kind of PCR model where you have invariant sequence around an ALU insert. And so you use those invariant sequences as PCR primers to amplify this region. If you've got the ALU insert, you may be 400 nucleotides would be plus ALU, and 100 nucleotides would be minus ALU. Now, that is really easy to resolve on a gel. The reason this is not used for standard DNA identification is that at each allele, you only have two possibilities. You either have it or you don't have it. So individuals are either going to be homozygous minus, homozygous positive, or heterozygous. With the CODIS system, at each allele site, there are sometimes half dozen, 10 individual copy number types found in the population. And so that gives you a much greater degree of variation at each individual spot. So that's what we have to say about that. All right, this along with the PowerPoint and a final or an exam sort of thing, a midterm is going to be coming your way as soon as I post it. Thanks for listening. Good luck, and I'll talk with you. Bye.